Hello, everyone, and welcome to today's webinar, The Journey from Neanderthal DNA to a Nobel Prize, How Dynabeads Magnetic Beads Contribute to Understanding Human Evolution. I am Shelley Mulock of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Erlend Ragnhildstveit. Erlend Ragnhildstveit has been at Thermo Fisher for 18 years and in his current role as R&D Leader for bead business in Norway since 2009. For a complete biography of today's speaker, please click their name in the presenter's window at the top right of your screen. Erlend, you may now begin your presentation.
Thank you, Shelley, for the nice introduction, and welcome. Good morning, good afternoon, and good evening to the audience. As Shelley said, so I lead R&D in Thermo Fisher here in Norway, and we are responsible for development of the magnetic Dynabeads. And with my interest in fossils, paleontology, and evolution in general, I'm really looking forward to present today's webinar. This webinar is, of course, referring to and also a tribute to Svante Pääbo, who is the last year's winner of the Nobel Prize in Physiology or Medicine for his contribution to understanding human evolution. And in my presentation, I will try to cover some of his work and key findings, show the importance Dynabeads drug Davidin and next-gen sequencing have played in advancing this field. And lastly, I want to introduce to you our newly launched Dynabead product that is optimized for target enrichment in the next-gen sequencing workflow. So, Svante Pääbo, the Nobel 2022 Nobel Prize winner, he's a Swedish geneticist and professor at both the Max Planck and the Karolinska Institute. He is regarded as one of the founders of paleogenetics and has worked extensively on the Neanderthal genome. The journey starts back in 1984, actually before, I should say, when he joined his mother to Egypt, got really interested in mummies. But the journey really started in 1984 when Pääbo is a graduate student at Uppsala. But he spends most of his free time, evenings and weekends, conducting strange experiments. He wants to extract DNA from ancient Egyptian mummies. So he sets out to replicate the mummification process. And he's doing that by putting small pieces of liver that he bought in the store into the oven in the lab. And he managed to isolate DNA, which suggests that dehydration via the mummification process might preserve genetic material. He then gets access to some real mummy specimens, and he managed to recover DNA from those. And they are nearly 2,000 years old, and he also managed to publish the results in Nature. This work led to two major realizations. One is that it's possible to extract DNA from very old samples, but also that DNA of that age is massively contaminated with other sources of DNA. He later realizes that some of the longer DNA sequence that was published probably came from human contamination and not from the mummy. So Pääbo becomes really a proponent for establishing methods and standards to isolate authentic DNA and not contaminants. And he becomes an exceptionally critical and careful scientist. In 1987, he joins a team in the US to work on the genome of extinct mammals. This results in sequencing parts of the woolly mammoth in 1994. 1990, Pebo returns back to Europe to focus on Neanderthal mitochondrial DNA. And in 2006, he announces, this is at the same time a next-gen sequencing instrument is coming in place. So he announces the plan to reconstruct the entire genome of Neanderthals. And in 2010, they successfully published a draft of the Neanderthal genome that most scientists thought impossible at that time. And actually, further in 2010, they successfully identify an unknown hominin based on genetic data alone from DNA extracted from a small finger bone found in the Denisova cave in Siberia. So they call this the new hominin Denisovans, and a high coverage genome was published in 2012. Now let's look at the workflow from bone to sequence. Extracting DNA from ancient samples is an extremely complex and challenging process. Over time, very old DNA degrades, becomes chemically modified, and is polluted with a large excess of DNA from microorganisms that colonized the tissue after the death of the body. In fact, ancient samples can contain 100,000 to 1 million times less endogenous DNA than fresh samples, with the consequence that even a tiny contamination of human DNA on a dust particle, for instance, in the lab, can overwhelm the experiment completely. So Pebo becomes a pioneer in developing standards and methods to isolate ancient DNA away from contamination. And Dynabeads, he's used Dynabeads from the very start, and they played an important role in separating out ancient both mitochondrial DNA or genomic DNA from background. The first step of the workflow involves grinding of a small piece of the bone. Then DNA is extracted, the pieces converted to a DNA library in order for the DNA to be sequenced. And the library step is typically where Dynabeads are used. In one variant shown here at the bottom, it's called a single-stranded library prep. And this is because when you do a double-stranded library prep, you trim off the ends. So you may end up with very small fragments because these samples are so degraded. So in order to get more of the longer sequences, you basically make also a library from each strand. And this is where Dynabeads strapped avidin comes handy in place to fish out through several steps. You can basically make libraries on each of the strands. You can actually also separate after amplification, do a size selection to only continue with the longest fragments. And this technique has improved the sequencing tenfold. So now more, what does this understanding of ancient DNA mean for the understanding of us as humans and human physiology? Many of you have probably done a heritage, an ancestry test to find or verify ancestral relationships. Some of these tests can also tell how many Neanderthal genes you have and how you differ in percentage Neanderthal compared to others. So why do most of us have Neanderthal genes in our genome? So studying genomes, we believe that Neanderthals, Denisovans, and today's humans, actually us, the modern humans, have some origin in Africa. And this goes quite far back. We're talking about maybe a million years. And that these ancestors then leave Africa somewhere 500,000 years ago. And in Western Eurasia, they evolve into Neanderthals, and in Eastern Eurasia to Denisovans. And then over time, the two populations have moved and overlapped at several times. For example, in southern Siberia, they have mixed and had children together. Then modern human evolves in Africa. So now we're talking about like 50 to 70,000 years ago. And they start to spread out of Africa to first mix with Neanderthals, and as they move east, to mix with Denisovans. Then Neanderthals and Denisovans die out, but kind of some genes from them are kept then in non-Africans. So with 1 to 2% Neanderthal contribution, and up to 5% or so Denisovan contribution in Oceania. And south of Sahara, there are 0% Neanderthal or Denisovan genes. Although probably also in Africa, the modern humans mix with other forms there. What does this genetic contribution of Neanderthals or Denisovan genes mean to us today, and how does it contribute to explain or define us as modern humans? A couple examples have some physiological or medical implications. One example involves the progesterone receptor, where a genetic variant exists outside Africa. This is the V660L. And it's almost certain that this variant is coming from Neanderthals. The variant is associated with preterm or premature birth, which is a risk to the child. So this is actually suggesting a selective disadvantage of this variant. However, the opposite is true. Over time, the frequency of this variant has instead increased, and data from biobanks are showing that the normal variant, the one that's not coming from Neanderthal, is associated with increased miscarriages and families with less siblings. So while the Neanderthal variant is associated with preterm birth, it's also protective against miscarriages, resulting in more live births. So this gene variant actually does contribute to increased fertility. And interestingly, studies have shown that if you give progesterone to women that had previously experienced miscarriages, you can substantially reduce the later miscarriages. Another example is related to the recent COVID pandemic. A big study showed that a very strong factor on chromosome 3, there are several genes there involved, that increase the risk of becoming very sick. The risk variant is coming from Neanderthals and exists in some humans today. A clinical picture in Sweden showed that if you were not a carrier of this variant and ended up in hospital, you had about 7% risk of dying. Whereas if you were a carrier, you had about a 13% risk of dying, so almost a doubling of your risk. When studying the frequency of this factor on chromosome 3, it is very frequent in South Asia, so India, Sri Lanka, with almost 50% carriers, while it's completely absent in East Asia, China and Japan. So it looks like it has been selected against in East Asia. Maybe they have experienced coronavirus before and been selected against, while it has some positive function in South Asia. And interestingly, it shows that this chromosome 3 region down-regulates CCR5, which is important for HIV infection. And I can actually show that if you carry this variant, you have about 26% less chance of getting HIV. So basically then, while the chromosome 3 locus gives high risk of dying from COVID, it is also associated with decreased risk of HIV infection. Third example is more defining us as modern humans and can explain why we are so different compared to earlier forms. For example, the stone tools from Neanderthals were very similar over the 400,000 years they existed, didn't really change much at all. And then suddenly when modern humans came, the technology changed rapidly. And art is another thing. While Neanderthals had very primitive art, it's difficult to see what it depicts. Modern human, you have much more figurative art. And another interesting thing is that while Neanderthals never crossed open water when they could not see land on the other side, modern humans start to spread all over the planet. And they moved to Australia, Madagascar, Americas. So why is this? And one interesting finding they have found is that they have found actually 3 genes involved in chromosomal segregation that results in a slower metaphase. This is the phase where the chromosomes align and they're going to be pulled apart. That takes longer time and seems to be more accurate in modern human variants compared to Neanderthal variants. Maybe then suggesting that modern humans have a better neural development and also a better functional brain. And you can also show that with the Neanderthal variants, you get more errors or fragments during this phase. So as you can see, studies of our closest relatives can contribute to better understand who we are as modern humans and give us some clues about human physiology. And as I mentioned, Dynabeads, and in particular Dynabeads streptavidin, has played an important role together with next-gen sequencing in this work. I'm therefore happy to introduce to you a new Dynabead that is specifically optimized for target enrichment in next-gen sequencing workflows. But first, I want to share some basics about the Dynabeads technology. Dynabeads are truly monosized, meaning completely spherical, and monodispersed, meaning they appear as single beads, and they are invented in Norway. They have become the gold standard of magnetic beads, especially in immunodiagnostic applications, and used in about 4 or 5 billion tests or several billion tests per year. They're also mentioned in more than 100,000 scientific publications. So while there exist many different vendors of magnetic particles, as you can, shown a few examples here, only not all of them have the quality of the Dynabeads that you see in picture A. What's important is really the uniform size, uniform magnetic distribution, make all the beads behave the same in experiments. While the biggest advantage is probably the extremely high batch-to-batch reproducibility. And this is very important. It secures same results time after time, year after year. So then our new Dynabead product is called Dynabeads streptavidin for target enrichment, and it's intended for enrichment of target DNA sequences made during the library prep as part of next-gen sequencing workflow. This is a 1 micron bead covalently coupled with streptavidin. And again, key benefits, as I said, reproducibility and sensitivity. And you can learn more about this product on our webpage. thermofisher.com. So then, if you look at where these beads are used in the next-gen sequencing workflow, after fragmentation of the genome and library preparation, you can isolate a subfraction of the genome by using specific probes that hybridize to your targets of interest. And since the probe is bioattenuated, it will bind to the streptavidin on the beads, and it can be pulled out of solution. You can do this in a direct way, where the bioattenuated probe is pre-coupled to the Dynabeads and then hybridized to the target, or in an indirect way, where the bioattenuated probe is first hybridized to the target and then enriched using the beads. Target enrichment is ideal, for instance, for exome sequencing or sequencing of a panel of genes, for instance, oncogenes, where instead of sequencing the entire genome, rather wants to achieve higher coverage of a smaller fraction of it. But still, this method can also be used in whole genome sequencing, as just shown in this ancient sequencing applications, to simply clean up the sample. Then I want to share some performance data of these beads with you. In the figure to the left, we compare these beads with other vendors, and the Dynabeads in red. And the red dotted line shows the threshold of the concentration of libraries required to go to the next step, to the sequencing. And Dynabeads here produce the highest concentration of final libraries. Samples below the line will not qualify and will not go further. Then in the figure to the right, then all the libraries, they're normalized to the same concentration for sequencing, and now the bars represent the quality of sequences produced. So the higher the bar is, the higher the quality. And again, you see a really good sequencing quality with the Dynabeads. In the last bottom figure, this shows how much sequence coverage you can achieve. Deeper coverage, more reads to build a consensus, generally increases the accuracy of results. And especially for clinical applications, the coverage is an important metric, as deep coverage is essential for rare variant calling. For instance, if you want to detect, let's say, a single point mutation in a cell that might be cancerous, you want to kind of distinguish that signal from noise. So the more coverage you have, the more likelihood to detect that small difference. So I think with that, I hope you learn a little bit more about this product, as well as how these beads are used in for ancient DNA applications. And I can just, my last slide, I want to thank you all for participating. Here is just some photo or pictures from my local, from my own fossil collection. You see a 50 million years old fish from, I think this is from Green River, like a piranha-like fish, and also at the right, this is a 1.8 million years old femur from a southern mammoth. So this is actually lived way before the woolly mammoth that I think most more people are more familiar with, but there existed also older ancestor versions of mammoths. So with that, thank you all, and I'm ready to move to live Q&A, and to you, Shelley.
Wonderful. Thank you, Erlend, for your informative presentation. We will now start the live Q&A portion of the webinar. Joining Erlend to help answer your questions today, we have Camila, Hannah, and Ivana. If you have a question you'd like to ask, please do so now. We'll answer as many questions as we have time for. So let's get started. Our first question today is, do you have any connection or collaboration with Pebo or his team?
Yeah, I wish we had, actually, since he's one of my favorite scientists. But at least not here at our site, we don't have a collaboration. They simply buy their beads from us.
Got it. Okay. All right. So the next question that we have then is, can you describe in more detail the workflow on how the Dynabeads are used in the ancient DNA field?
Yes, sure. Actually, they're used in many different ways. Maybe one thing is what I just showed. When you in this library prep, you have one of the adapters biotinylated, and you can simply then with the beads pull out the library fragments. Or as I showed you with the single-stranded libraries, you use there are different tricks where you use biotinylation and different oligos to basically form a double-stranded library fragment. And also use the Dynabead streptavidin. You can simply, because you get a mix of very small fragments and longer fragments, and if you want to enrich for only the longest one, is the Dynabeads are really useful because you can simply heat the nature of one strand and do a size selection on a gel to continue. But also I mentioned the mitochondrial DNA work they did. Then they used basically mitochondrial gene sequences as bait to pull out the mitochondrial genes or ancient mitochondrial genes from the background of bacteria. So that's another application they have used.
Very interesting. Thank you so much. Okay, another question that we have is the product specific for target enrichment in the NGS workflow, or can it be used in more genetic ways?
Yeah, so this product is intended for kind of optimized or intended for this next-gen sequencing workflow. But we also have other similar streptavidin products that more generic use.
I see. Okay. The next question that we have is, I can see how the person who asked the question said, I can see how Dynabeads has a clear advantage over competing beads. They're curious about cost comparison and if there's a trade-off between cost, product availability, accuracy, and efficiency, those kinds of things.
I don't know if Hannah, you want to...
Cost is always a difficult question to answer because it's so, regionally dependent. But what we can say with the accuracy is that we do boast high reproducibility in our products. So especially when you want a workflow to move towards the NGS, where it's crucial to have robustness in your workflow to have a successful result. That's where you see the huge Dynabead advantage. You get the same answers or same quality again and again.
Yeah, I think that's an important aspect. In one way we can look at it is that we invest a lot into the quality of the Dynabeads. but then in the what you get back is more successful than or successful experiments. This goes back to the batch-to-batch reproducibility. You can expect same results always. Also, we kind of build in this this extra quality build in will also save costs with the customer, not to mention frustrations.
Yes, absolutely. Okay, wonderful. Thank you. The next question that we have for the team is how forensic science and developmental biology are intertwined. Yes, how are they intertwined is the question.
Forensic science and developmental biology. Yeah, that's maybe, I don't know if Hanna or Camilla want to give it a shot.
I'm not exactly sure we understand the question correctly.
Yeah, not sure. Maybe.
Well, what we can do is always push that one through, and maybe have your team take a look at it. And all the questions that come through will be sent to you. So
but there is a but there is a point, I would say, with forensics studies. I mean, if they mean more like ancient DNA versus forensics, that's maybe that's what the question is about.
Okay.
That's very interesting, because then you also have tiny amounts of DNA in a background of a lot of contaminants, and how to get that, fish out that with the quality. I think that's a very good question. And I see that they are actually intertwined in the sense that you have to be particularly careful to enrich for the right material away from the vast excess of background. Yeah.
There's also an overlap in the type of challenging samples between forensic and this type of ancient.
Yeah, a lot of overlap exactly in the challenge with the samples from forensics versus this ancient DNA samples.
Good, excellent. Okay. Another question that we have is Dynabeads, do they help to isolate the cell-free DNA from liquid biopsy?
Indeed, that is absolutely a perfect match for this Dynabeads functionality. It is developed precisely for this type of precision medicine workflow, where you want to enrich for a specific panel of oncotargets, moving into the NGS type readouts.
Okay, wonderful. Well, as we sort of wait for a couple more questions to come in, I just wanted to see if your team had any thoughts that you wanted to share in the meantime.
No, I think we kind of showed a little bit. Nice thing about the Dynabeads and the streptavidin beads is also the high versatility. You basically, as long as you have something biotinulated, you can basically use these beads for anything. Still, when we are saying that the new product is kind of intended and optimized for the next-gen workflow, we can, of course, use it for other things, but it's more that we optimized everything specifically for that workflow.
Wonderful.
It doesn't mean that this bead does not work in other workflows.
Sure. Okay, well, thank you again, Erlend, for your time today and for your important research. We'd like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. So before we go, I'd like to thank the audience as well for joining us today and for their interesting questions. And questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So thank you so much again, and until next time.
Thank you. Bye-bye.